Hey everyone, welcome to Professor Long's Lectures in Microbiology. These videos are intended for use by students who are enrolled in my microbiology class, Biology 2420, which is microbiology for the Allied Health Sciences, here at Del Mar College. If anyone out there in YouTube land finds these helpful, and if you're a my student and find them helpful, by all means, please give me a like and subscribe to my channel so that any new videos that pop up, you'll be notified, or provide me some feedback, okay? Now, your instructor may go into different detail, but this is the information I want my students to know, the stuff that I emphasize. So what we're going to be doing now in this lecture is we're going to be talking about the history of microbiology, at least the origins of the study of microbes and microbiology. We're going to go through a list of scientists that made great contributions. This is not by any means a complete list or a detailed list. It's just a general broad overview of some of the major hitters. I may leave one or two out that some people like to talk about, and I may include some that others don't talk about. So my list is for my class that may be incomplete for other classes. So please learn all those scientists and their major contributions to microbiology. The dates are somewhat important. I'm not always going to go over the dates in order, but nonetheless, let's start talking about some of these guys. Now, prior to the first few guys that I'm going to talk about, there was a lot of ideas of where life came from. And one of those ideas was called spontaneous generation. And I want to mention this to you because we're going to talk about it a few times. So, spontaneous generation means that things would come about spontaneously. And this was the belief that non-living particles or substances could assemble spontaneously and form life or form a living organism. And people would look at a rotting corpse or a rotting organism or just some old stuff sitting around like a bucket of fluids or horse dung or whatever it was, and then eventually they would see little creatures living in it, like maggots growing and crawling around, and they would go, oh my goodness, all that stuff has some compounds in it that when they mix just right, forms life. And they thought that life sort of evolved spontaneously, and they called it spontaneous generation. And it really is the theory or the belief that non-living substances and particles can assemble to form living organisms or life. And this was believed, for a long time. Prior to that, some ancient peoples and tribes and things would think that the way that things happened was because of voodoos or curses or the gods or whatever it was. We would sacrifice things to make it rain, and there's all these crazy ideas that existed that didn't seem crazy at the time. Today they might seem so to many of us, although some of them are still practiced in some parts of the world. Now, there's a list of guys or characters, scientists that came about that made some major contributions to dispelling these ideas of spontaneous generation and what life was, how it formed, and where it came from. So I'm going to talk about some of these guys and the major hitters. And one of the first ones that we always talk about in microbiology is a guy named Robert Koch. Okay? Now, this guy came up with the cell theory. He was the father of what we call the cell theory of life. And essentially what he did was he had a very crude microscope. He crafted a lens, and as he was looking through this piece of glass that he noticed caused things to appear larger, basically a piece of glass that he used like a microscope, what he realized is that he cut up some cork. And as he looked at the cork, under this thing, he saw inside of it that it was broken up in all these small little structures that he called a cell. Like the cell that a monk would live in, the room that a monk would live in, or like a jail cell. And he noticed that the cork was made up of all of these units, and as he looked at other things, he saw the similar layout. And so he described them as cells. And he came up with the idea of what we call the cell theory of living organisms, that all living structures are made up of cells. Okay. Now, there's another guy that in the 1670s, somewhere around 1673, this guy named Anton van Leeuwenhoek. He's a Dutch guy, has a unique spelling to his name. But Anton van Leeuwenhoek was the first to see or observe microorganisms. Turns out that this guy was a carpet merchant who devised little tiny microscopes, little handheld pieces of wood with a little lens, a round lens in them, and he would examine the fibers in carpets to see how finely they were made and things as people were trading these things. And he also was the first one to see little tiny microorganisms, or what we call microbes. He called them animalcules, like molecules that had animal-like characteristics or living characteristics. And he was the first guy to see, observe, and draw these microorganisms and describe them. And he went through a whole bunch. And he did this for about 50 years, from about 1673 to about 1723. He spent, half a century describing all these organisms. Okay, so he was one of the major players in that he came up with the idea of these microbes existing. Again, he called them animalcules. Now, in about 1668, there was a guy named Francesco Redi. An Italian. Now, Francesco Redi's major contribution was he was the first person to challenge the idea of spontaneous generation, or at least he's the first to publish anything on it. He was the first guy to say, listen, these organisms don't just spontaneously generate, that all life must come from other life, okay? That all living things come from pre-existing things. And basically what he said was that he noticed that every time flies landed on something that was rotting, later on he saw maggots. And he said it must be that the flies lay some kind of egg, like a chicken lays an egg and it gives birth to a chick. And so he challenged the idea of spontaneous generation. That was kind of controversial at his time. It was a big deal. He was one of the first to buck the trend, and the whole world went, oh no, you can't do that. Now, in about, somewhere around 1745 or so, there was a guy named John Needham, an Englishman. John Needham was not only a scientist, he actually happened to be a Catholic priest. And you would think people who were strongly religious and opposed things like evolution would be extremely opposed to this idea, but he kind of kept spontaneous generation alive. As a theory, he sort of prolonged the idea of spontaneous generation. And so he did some experiments that he thought disproved the idea that the spontaneous generation could not be true. And essentially what he did was he would boil some meat, or he would boil some meat broth and let it sit out, and he would come back later and he would see life living in it. And he said, you see, when you boil something, you kind of sterilize it and you kill anything in it that could be alive. You kill all the cells under any living part of it, and I'm still getting life coming out of this. And so it must be spontaneous generation. And so he sort of prolonged this concept of spontaneous generation. He would see maggots growing in boiled broth and boiled meat. The problem was his experimental technique was flawed in that he did not completely prevent flies from getting in. He just never observed the flies laying their eggs, causing maggots to grow. Okay? So, now, shortly after this guy, it took about 20 years, okay? And of course, all of what we called, all of this stuff was happening in a period that we refer to as the Renaissance. And you don't need to know this for my class, but the term naissance means birth. And the Renaissance means the rebirth of science and the rebirth of knowledge. So we had gone through what were considered the Dark Ages. In the Dark Ages, there wasn't a whole lot of learning and advancement. Everybody just kind of lived in the same world, kind of ignorant. The only people that could get educated were people that read Latin, that went to the church and studied under the Roman Catholic Church, and all you did was translate Bibles. During the Renaissance or Rebirth, people began thinking for themselves and studying and writing things that very often were considered sacrilege, many of which these guys did. So a lot of this was kind of in that first Renaissance period where the rebirth of knowledge started coming around. Well, this one guy named Lazzaro Spallanzani, another Italian, I believe, he was the first to come around and say, look, I redid Needham's experiments, but his technique was flawed. So essentially his major contribution is that he did the experiments that disproved spontaneous generation. And essentially he came in and he said, look, Needham's experiments have to be flawed. He disproved John Needham's idea by saying, look, you boiled all this stuff, but you still didn't prevent the flies from getting in. So he would boil meat and completely seal it in a jar where no flies could get in, and nothing grew. He did the same thing with broth, and he said, you see, his technique was flawed. If we completely seal the stuff off and prevent other living organisms from coming in and laying eggs, then you don't get any life out of it. It just rots, and that's it. It just sits there. And so he completely killed the idea of spontaneous generation, which I think is interesting that a Catholic priest kept the idea that life evolved from non-life. But Lazzaro Spallanzani essentially redid Needham's experiments with better technique and shut that down. So that's his major contribution, is he disproved spontaneous generation by redoing Needham's experiments. Now, also, somewhere around this period, in the 1750s, one of the big publications by this guy was in like 1758, was a scientist named Edward Jenner. Now, one thing we talk about Jenner when we talk about him is we say that he is the father of immunology, or one of the fathers of immunology. Okay? He created what we consider to be one of the first vaccines. Now, quite honestly, some of this stuff was happening in China, but he was the first guy to really write about it in scientific literature and show that vaccines work. And the term vaccine comes from the term vaca, which also some of you, if you speak Spanish, know vaca means cow. Okay? And what basically he noticed was this. There was a disease spreading all over Europe called smallpox, a plague, and it was wiping out people. If you got sick with smallpox, you were going to die an ugly, virulent death. And it was wiping out large parts of the population. It was killing cattle, but it was also, no, it wasn't killing cattle, it was killing humans at an alarming rate. And it was a disease that was only affecting humans. Well, he also noticed from a group of ladies called milkmaids that would go out and they would milk cows and come back with buckets of milk, that they contracted a disease that cows had. The cows would get little pustules on them that would, break up like little sores filled with pus, that was formed from what we call cowpox, like a pock mark on something. And so the cows had these little sores all over them that they called cowpox. And the milkmaids would pick up cowpox. They would get these little sores on them that were similar to smallpox. But what he noticed was the milkmaids that were infected with cowpox never got smallpox. And he said there's something in this pus of the cow that is causing them to get sick and get a fever and get these little pock marks on them. But then they never get smallpox and die. So somehow their body is reacting to something in the cowpox that also prevents them from getting cowpox again in the future. They would only get it once, but they also don't get smallpox. He didn't know what it was, but he knew there was an association there from these milkmaids. And so what he did was he would take a needle and stick it in these little pock marks on cows with pus and infect people with them. And everyone that he inoculated with cowpox was safe from smallpox. And so that term vaccine comes from the term vaca, from cow, and that was the first vaccine, or at least one of the first documented vaccines in Europe. And so Edward Jenner used cowpox to inoculate people to prevent them from getting smallpox, and eventually smallpox died out and was not such a big deal. So we call him the father of immunology, okay? Now, also, it was about 100 years later that this guy named Rudolf Virchow, or some people say Virchow. I think he was of some kind of Russian descent, okay? Now, Virchow's major contribution was that he helped solidify what we call the cell theory of disease, okay? So Robert Hooke was the first one to come up with the idea of the cell theory of organisms, that all organisms are made up of cells. But it was Virchow that solidified what we call the cell theory of, or he solidified the cell theory, and he came up with the idea that of what we call the cell theory of pathology, or the cell theories of disease. So his major contribution is he solidified what we call the cell theory. Basically what that says is all cells come from pre-existing cells. He also said that all organisms get sick from other ill organisms. And basically, that all organisms get sick at the cellular level. Man, my penmanship near the bottom of the board is terrible. Essentially, this is what he said. All organisms get sick at the cellular level. So prior to this idea, people thought when you got sick, the whole being of you was ill. And he said, no, first of all, the cell theory is true, that all cells come from pre-existing cells, and that when an animal gets sick, it's sick at the cellular level. And certain group of cells can become infected with something that causes the whole organism to feel bad. And so he came up with the cell theory of disease as well, or the cell theory of pathology. So he helped solidify the cell theory that all cells come from pre-existing cells, and he solidified the cell theory, or came up with the cell theory of disease, or the cell theory of pathology, and that the organisms get sick at the cellular level, okay? So now, after this, or around the same time, one of the major players in microbiology, and this guy is huge. If you don't know this guy, then there's a problem. Louis Pasteur. Now his work occurred over a long period of time, but one of his major publications was around 1861. And basically what Louis Pasteur came up with is that it's microbes in the air, water, or soil that cause disease. Okay? And he basically said that microbes in the air can contaminate other substances. That there's microbes blowing around in the air that can land in a bucket of water or a bucket of broth and cause it to spoil or sour or become diseased. And that there's microbes in the soil that if we dump them in water or in a solution, that can cause other microbes to grow. Okay? So basically it's microbes in the air, water, or soil that can cause disease, or he said contaminate sterile solutions. So one of the things he did was he took broth and he boiled it and he sealed it off so that he knew it was sterile. Nothing would grow in it. And then if he took the cover off and left it exposed to the air, he would notice a little film of the stuff growing in it, which were bacteria. And he said, look, these little living organisms, these bacteria that are growing in here, came from the air. They're blowing around in the air and they're on the surface of things. So if you touch it, if you mix soil with it, if you mix other solutions, there's microbes that are in the air, in solutions, and in soil that can cause or contaminate sterile solutions and cause the disease process or cause things to become contaminated. And so we call this the germ theory of disease. And he was the one to first come up with it and publish papers on it. And he said it's germs causing disease and contaminating things that are otherwise sterile or not contaminated. Okay? He demonstrated that microbes can live in the air, microbes live in water and other solutions, and that microbes can live in soil. That they lived in inorganic material sometimes, or that they lived in non-living things. And when mixed with certain living situations or certain chemicals, we could contaminate them, and therefore it is these little germs or microbes that are in these non-living substances that can end up causing diseases. Another thing that he showed was that heat can kill microbes, or most of them, a process that we call pasteurization, after Louis Pasteur. To pasteurize something means you pass it through something that's really, really, really hot so that you kill all the microbes in it. At least that was one type of pasteurization. And so people started heating things up before they would seal them in jars. They would kill the microbes, and therefore things would last a lot longer and wouldn't spoil as quickly, and you wouldn't get sick from it. Okay? So, he also was the one that showed that microbes can cause spoiling of solutions and spoiling of food and spoiling of milk and other things. And so he came up with pasteurizing milk and pasteurizing other substances so that they wouldn't spoil as rapidly. So Louis Pasteur was there's a lot of other major contributions that a couple of these guys gave, but these were the big ones. He came up with pasteurization, the heat killing of microbes in air or in solution, so that they cannot cause contamination or spoilage. And he also came up with the idea that it's germs and microbes causing diseases. Now, it wasn't 100% solidified, but he was the idea that started this concept. Now, one of the major players shortly after him, and this guy is also a huge one. I'm going to put 3 little asterisks next to these 2 guys. You better know their contributions. You should know all of them, but if you miss these 2, you're going to miss this question on every standardized test you take from here on out about this stuff. There's another guy named Heinrich Robert Koch, and he actually Heinrich Hermann Robert Koch, but most people know them as Robert Koch. Or some people pronounce it Koch, and some people pronounce it Koch. Koch's postulates, Koch's postulates, or Koch's postulates. I learned it as Robert Koch, K-O-C-H. Now, he created or came up with what we call Koch's postulates. He postulated or hypothesized that this happened, okay? And he also helped create sort of the germ theory of disease. Louis Pasteur sort of gave the idea, but he solidified the germ theory of disease for sure. And he was the one to demonstrate it, where Pasteur came up with the idea. This guy demonstrated the germ theories of disease and solidified it as truth in science. Now, one of the first things that he discovered, by the way, was Bacillus anthracis. So this is the genus and species of a bacterium. Bacillus anthracis, he said, is a bacterium that causes anthrax. And anthrax is an infection that can kill cattle and cause humans to get very, very sick and kill humans as well. And he actually discovered that it was a specific organism that he named Bacillus anthracis that caused anthrax. And he went through all the taxonomy and got down to this genus and species and said, this is the germ causing this specific disease, and I can prove it. So it solidified this germ theory of disease. He also figured out what caused tuberculosis and, what's the other disorder or disease? Let me check some notes real quick. I can't think, I'm drawing a blank right off the top of my head. It was three major ones. Cholera. He's the one that figured out what caused anthrax, tuberculosis, and cholera, as well as some other diseases, and he proved that specific organisms cause specific diseases, or specific germs cause specific diseases, and so he solidified or proved the germ theory of disease. Now, essentially what you need to know, another thing you need to know is you need to know what Koch's postulates are. So I'm going to write them out. And they're written a little bit differently from textbook to textbook, but they essentially say the same thing. So these are Koch's postulates. Again, this is stuff you need to know. This is one of the central themes in all of microbiology. If you don't know this, it's on your final exam, it's on the first test. You should at least get a 2 or a 4 on the first test. You should know Pasteur and you should know Koch and what their major contributions were. Okay? So I'm going to erase his name here. And we're going to continue Koch's postulates. Okay? So now, basically what Koch said was this. Okay? That whenever we find a disease organism, that we should be able to identify a microbe that is found in the diseased organisms and not the non-diseased organisms. So the first of the 4 postulates is a specific microbe. And again, this is somewhat of a it's a sort of a rewording or a different way of saying it, but again, different books have it written out slightly differently. But basically, a specific microbe can be found in diseased organisms and not the healthy ones. So, for example, if there's a cow that has anthrax, I should be able to see an organism that I can find in the diseased organisms, the ones that exhibit anthrax, and not the ones that are free of anthrax, that are fully healthy. Okay? And he said we should be able to isolate or the disease-causing microbe, which is also called a pathogen, can be isolated from a diseased organism. Now, whatever the infectious agent is, whatever the microbe is that's causing the disease, you should be able to isolate that organism or isolate it from a diseased organism, and you should grow it in culture. He wasn't the guy that came up with agar. Agar meaning, this is spelled A-G-A-R. Agar is the medium that we'll see in lab that we can grow bacteria on. It has some of the nutrients and stuff, and it's semi-solid at room temperature. And so he was able to grow organisms in agar and isolate certain organisms and grow them on plates and petri dishes and show that specific organisms could live on this agar and could be isolated from other organisms. And essentially what he did was he would take a sick organism, like a cow, for example, and he could isolate the pathogen or the disease-causing organism or the infectious agent, all the same thing. They could be isolated from a diseased organism and grow it in culture. So if you're sick, we should be able to swab your nose, and if there's some bacterium causing an infection, like if you have a sore throat from Streptococcus bacteria, strep throat, we should be able to swab your throat and show that that bacterium is there. It can be grown in culture. Okay. The next thing he decided, or he postulated, was the cultured pathogen can infect a healthy organism. So, essentially, I can take an organism, I can find an organism that is diseased, I can isolate the pathogen or infectious agent from a diseased organism that is not necessarily present in the healthy ones, and that can be cultured. So I can swab someone who's healthy and rub the same agar, and then take another tube of the same agar, swab someone's throat who's got strep throat, and I will see the bacterium grow on this one and not on the healthy individual's culture. So essentially, that a specific microbe can be found in diseased organisms and not the healthy ones. That disease-causing microbe or organism, the pathogen, could be isolated from a diseased organism and grown in culture, and that the cultured pathogen can now be used to infect an otherwise healthy individual. So if I take a cow that has a specific disease, like anthrax, and I have a cow that's free from anthrax, that it must be a microbe causing that organism, and we can find it in there. I can take that microbe and isolate it and grow it in culture. I can take the cultured microbe and inoculate a healthy organism, and that organism will get infected. It will get the disease. And finally, the same pathogen can be isolated from the inoculated animal. Now I'm paraphrasing. That's not exactly the real detailed scientific terminology, but it is the same terminology. It's just, written in a little bit easier to understand terminology here. The same concepts put in different language. So a specific microbe can be found in diseased organisms and not healthy ones. That disease-causing microbe or pathogen can be isolated from a diseased organism and grown in culture. That the cultured pathogen can be used to infect a healthy organism and produce the same disease. I left that out. And then finally, that we can take that newly infected organism, and when that pathogen can be isolated from the inoculated animal, and we can show that growing in culture again. So I can take a sick animal, I can swab it and isolate the disease-causing organism in a culture. I can take this cultured infectious agent and introduce it into a healthy organism, and it will produce the same disease that the other diseased organism had. And then I can take this newly diseased organism and I can isolate the same pathogen, thus proving it was this organism that made this, it was a particular microbe that made this organism sick. I can isolate the microbe, grow it in culture. I can take the same microbe and cause a new healthy organism to get infected, producing the exact same disease. And then I can isolate that same microbe from this diseased organism, proving that I infected it, and this is what caused that disease. So that was the germ theory of disease, solidified by Robert Koch, and he postulated these four things, these four steps. Okay, so you should know Koch's postulates. You can look them up in the textbook or look them up on Wikipedia or wherever you look, and they're written slightly differently, but this is what it essentially says. Okay. Now, one of the other major players is Joseph Lister. So I hope you have Koch's postulates down. I'm going to erase them, and the last guy we're going to talk about is Joseph Lister. Now, sort of the father of disinfection and aseptic technique. Now, and this was somewhere around the 1860s. So I think it's around 1865 that he first published his papers. But he basically, he used a compound that we call phenol to disinfect his hands, to wash his hands and clean them to kill any infectious agents. And so he came up with the idea of disinfecting things using chemicals. So he's the father of disinfection. Come on, Bob. Can't write very well, sorry. He's the father of disinfection and what we call aseptic technique. And he used a substance called phenol to disinfect things. And essentially he showed that not only can heat kill microbes, but there are certain chemicals or compounds that can kill microbes, and one of them was this stuff called phenol. Now, one of the substances that we use to kill the bacteria in our mouth is called Listerine, and it gets its name from Joseph Lister. So, essentially, he was the first one to show that I can transfer disease from one person to another or from one thing to another by carrying that disease on my hands or on my body. And if I were to just wash my hands with this stuff called phenol, and by the way, phenol is also called carbolic acid, which is kind of a tough acid. It's just stuff that's really not good for your skin in high concentration. But he basically showed carbolic acid, or what we call phenol, would kill the microbes that could be transferred from one substance to another. So you could touch something else, and transfer the germs. But if I'm touching something that's contaminated with germs, and I wash my hands with carbolic acid or phenol, and then I touch something else, then it killed the microbes, and I wasn't transferring the germs. And we call that asepsis. When we talk about sepsis, or something being septic, that means it's contaminated. Asepsis means lack of contamination or not contaminated. So he came up with aseptic technique, and this led to a revolution in surgery, because before him, surgeons would go into a room, they wouldn't even wash their hands, they would wear the same apron, they would do a surgery covered in blood, they would wear the same apron to the next surgery, they would go in with blood on their hands and touch the blood from one patient to another. And if someone had an infection, they didn't realize they were transferring infections, and they didn't realize that it was these infections that were often killing their patients. And Joseph Lister was the first guy to come in and say, listen, we need to use sterile or aseptic technique. You need to change your apron. You need to wash your hands. You need to wash the aprons of your surgical gowns and not carry the contaminant from person to person. Okay? So he basically revolutionized surgery by stopping the spread of disease-causing organisms that resulted in a lot of surgeries ending in death after infection. All right? So, now in my opinion, for now, that is enough of these guys. There are some other major players, but these are the ones I want you to know who they are, what their major contributions are to science and to microbiology. Now, roughly what we call the Golden Age of Microbiology is from around the 1840s, 1850s to the early 1900s. So from about the 1850s into the 1900s, we call this the Golden Age of Microbiology. And whenever we talk about something being the golden age of something, we mean this is where it really flourished and became a big deal. Okay? So, essentially, what was happening during this period is we were starting to discover all of the disease-causing organisms. we could prove that specific organisms cause specific diseases, and then we could begin to start to treat these diseases in certain ways with aseptic or sterile technique, and also using other compounds or processes like heat pasteurization to stop the spread of microbes so that things didn't spoil or become contaminated or infected. Now, this is where it really flourished, and a lot of information came out that became the real foundational period of microbiology. A number of things that this allowed us to do was to diagnose and to treat a lot of diseases on the cellular level and on the biological level by killing and stamping out microbes or stopping their spread. Now, a few things that I want to discuss before we wrap this up. When it comes to treating diseases, there's different ways to treat diseases caused by microbes. And this is just sort of a basic general introduction. But one of these is by chemotherapy. Chemotherapy is the use of chemicals to treat disease. We often hear of chemotherapy when we talk about the chemicals that are used to treat cancer. But chemotherapy really is the use of chemicals to treat any disease, any disease process. Now, some of the chemicals that can be used, one group is called antibiotics. Now, anti means not, and bio means life. So antibiotics are drugs used to kill microbes, and particularly bacteria. Although some things are classified as antibiotics that treat other microbes or other disease situations, but one way that we can describe antibiotics is that we can say that they're usually produced by other microbes, by plants, or synthetically. Now, the first treatments for diseases, the chemotherapy agents, were used in the 1800s by the conquistadors from Spain, but what happened was the conquistadors from Spain, as they came into the New World, particularly the southeastern parts of the U.S., from Florida all the way through Louisiana and Texas, actually all the way through California, the Spaniards came in and even into Central America, into Mexico, Central America, and South America, wherever they ended up, they decided they were going to claim this territory in the name of the King of Spain, and that because that they had this religious morality, that they were going to turn everyone into Christians, and they basically, conquered everything, hence the name conquistadors. Well, the Spanish conquistadors realized that when they were going through the jungles of these areas, many of them would contract malaria. They didn't know where it came from. Turns out it comes from certain mosquitoes spreading a microbe. But they realized that a lot of the indigenous peoples used a certain tree bark to treat the symptoms of malaria and make the disease go away. And we now know that there's a substance called quinine in tree bark, but they use the tree bark of a specific tree to treat malaria. Now, that was sort of our first documented use of chemotherapeutic agents, and although there were some going on in China and India and parts of the East as well. Now, this was one of the first known uses, was in the early 1800s, right? The use of quinine from tree bark to treat malaria. But in, 1910, I think it was 1910, or about 1910, a guy named Ehrlich actually used Paul Ehrlich. I couldn't think of his first name. Paul Ehrlich uses stuff called arsenic. arsenic is really a poison if it's taken in too high a concentration, but he used arsenic to treat syphilis. Now, syphilis was a sexually transmitted disease that would start to eat up the body and parts of the nervous system and basically kill people if it went untreated. So he was the first person to use arsenic to treat syphilis and showed that, you know, chemotherapy is real. And this started sort of the massive drive to start discovering new compounds to treat diseases. And people were trying all sorts of stuff, some of which were really toxic. It was in 1928, and he at least, I think, published his paper, but a guy named Alexander Fleming, he discovered penicillin. Now, basically penicillin, or the drug that we now know as penicillin, was isolated from penicillium, and I'm drawing a blank on how to spell correctly the species, and I should know. Chrysogenum. I always misspell this too. There's so many of these bacterial, or so many of these genus and species names, and some of them are really hard to spell and hard to remember over time. So, but Penicillium chrysogenum produced a compound that killed certain bacteria. And so what he realized is that this mold, basically here's what happened. He would have a petri dish with agar that would be covered with bacteria. And what he noticed was on some of his plates, a couple of the plates got contaminated with the mold. And with the area where this fuzzy looking mold was growing, the bacteria wouldn't grow anywhere near that. And he saw these empty spaces on the plates, and he theorized, well, maybe this mold is releasing some compound that's preventing the bacterial growth. And when he isolated that compound, it later became known as what we call penicillin, which is a major antibiotic still used today. Okay? And of course, in the 1940s, during World War II, when soldiers were being shot up and worked on in the field, they started using this compound because they could isolate it from these plates that were where we were growing the mold. They could isolate the compound that was killing the bacteria from growing, and then they would treat these wounds with this antibiotic, which would stop infections from wounded soldiers, and many more soldiers survived during World War II due to the advent of antibiotics and the use of penicillin. Now today we have many more antibiotics that we can use. You're going to learn some of those later in the semester. So now, that's somewhat of a brief history of microbiology. Let me check the time on my video. Yeah, we're getting pretty long. I'm trying to keep these under an hour. But man, let me tell you, microbiology is a very long topic with a lot of information. And again, I could probably do this very rapidly by showing you a bunch of PowerPoints and flying through information. But the goal is for me to help you learn. So, the last thing I want to cover today in this lecture, and we can wrap up the first chapter of the book, is certain topics within the heading of microbiology, or subjects under the title of microbiology. And you know, these are pretty easy to learn. The first one, when we talk about topics, and there's more than this, but these are the major topics in microbiology. Okay. Let me make sure, so let me get behind my camera and make sure that this color shows up, because some of the colors don't show up very well on the board. Okay. But some subtopics in microbiology. One of them is called bacteriology. Well, that's pretty easy. If you know ology means the study of, this is the study of bacteria. And sometimes you'll see this word, bacterium. Bacterium means one type of bacteria, or singular, basically. So Streptococcus pneumoniae is one bacterium. Klebsiella pneumoniae is another type of bacterium. There's Streptococcus aureus, or Staphylococcus aureus, I should say. Those are three different bacteria. Bacteria is collective or plural. Bacterium is singular. So you might see it as bacterium sometimes or bacteria. But bacteriology is the study of bacteria. Mycology is the study of fungi. Okay? So funguses can cause disease, but some of them are very beneficial as well. And so whenever we talk about studying fungi only, we talk about mycology. Now, virology is obviously the study of viruses. Parasitology, is the study of parasites and the diseases caused by parasites, the parasite life cycle. We talk about the life cycle and the processes of these organisms and stuff. So immunology is the study of immunity. How do organisms become immune to things? It's the study of the immune system. Our body has immune cells, white blood cells, that have the ability to recognize foreign cells and attack them and prevent infections. So if you get exposed to a low level of something, your body might be able to prevent the disease, even though you have it on your body or in your body and you're carrying it or infected with it. You're keeping it at such low levels that it doesn't cause any disease process, and eventually your white blood cells and your immune system can wipe it out. Now, when enough of the organism enters your body to overwhelm your immune system or to circumvent your immune system to get around it, and it starts causing a disease, that's when you need health care and chemotherapy or the use of agents to help fight the infection. Okay? Now, while we're talking about all of this, one way that we can fight infections, or at least study bacteria and viruses and some of these microorganisms, is what we call recombinant DNA technology. This is the use of technological means to alter or recombine genetic material. Essentially, this is what happens. Viruses carry some enzymes in them, and we're going to talk about what enzymes are and how they function in more detail. But essentially, there's these proteins called enzymes, and a specific group of enzymes called restriction enzymes. They can find a series of bases in DNA, and they can cut the DNA in this particular area. And so we can chop up DNA into smaller fragments. And then we can rearrange the DNA. If I have an enzyme that recognizes a series of bases, and when they cut, these bases actually match and overlap, I can recombine the DNA into a new sort of genetic makeup. And the technology used, the techniques used to alter and recombine genetic material is called recombinant DNA technology. And so, essentially, we can take all the DNA of an organism, called its genome. This is the total complement, or the total genetic makeup, we won't use that term, of an organism. It is the entire genetic sequence. What are all the bases within an organism? So simple organisms like bacteria and viruses, and even some very complex organisms, we've been able to figure out technological ways to chop up the DNA and sequence what are the order of bases, and not only figure out what is the entire series of bases that makes up all the DNA or RNA in an organism. but we also now can recombine it and try to create new organisms, or at least genetically modify those organisms. Okay, and the whole study of DNA and the function of DNA and how DNA is converted into RNA and proteins is called molecular biology. What am I writing here? Molecular biology is basically the study of DNA structure and its function. And I like to throw RNA in there because it's a part of it. So, what is the order? The study of the order of bases in an organism, and its DNA and its RNA, and how those function to produce proteins and cause an organism to function a certain way, is called molecular biology. Cell biology is studying the biology of a cell. Molecular biology is really studying those molecules, particularly the DNA and RNA, and sometimes the proteins within the molecule, and how they function. And if we're going to do this, we need to know the genome of an organism. If I know what genes it has, I know what proteins it can produce. I also know what RNA must be there, so that I can understand how this organism is using its DNA and RNA to produce proteins and function. So that's all that molecular biology is the study of the DNA and RNA structure and function and how proteins are produced. In order to do that, we must know the genome. And once we start manipulating this genome with technology to recombine it into new orders of genes, it's called recombinant DNA technology. Essentially, this is what can happen. If I know a restriction enzyme can cut a series of bases, and then I can literally take those two cut pieces and stick them back together. What if I could cut a piece of DNA open and take another piece of DNA cut with those same enzymes and stick it in between these two cut ends, and now I've recombined two pieces of DNA. I can cut a gene out of one organism, I can cut the DNA of another organism and insert the gene into that organism's DNA. I can recombine the genetic material, and now when this organism is producing its proteins, it's also going to make the protein that this new gene is coding for. And we're going to talk about recombinant DNA technology and how it occurs in great detail. So, now you know a little bit about the history of microbiology, who some of the major hitters were. You should know kind of the year about where they did it and what their major contributions are. You should know the topics in microbiology and these definitions. So I hope during this process you learned something. I hope you had as much fun as I did while we were going through it. And then in the next video, we're going to start doing some of the chemistry in this class, which is a really interesting and fascinating topic for me. You cannot enjoy biology, especially microbiology, without knowing some chemistry. So I hope you learned something. I hope you had as much fun as I did. I'll see you in the next video. Thanks for watching.